что, тогда начинаем. Большое спасибо, Марина, что вы сегодня согласились нам прочитать лекцию. Марина работает в Университете Калифорнии в Сан-Франциско. У нее есть своя лаборатория, которая называется Sirota Lab, и они занимаются использованием открытых клинических данных для разработки новых инструментов с целью в конечном счете получить лекарство. А я почему-то сейчас себя слышу. Так должно быть?
Мы тебя слышим.
Ну хорошо. Главное, чтобы лектору не было слышно в процессе чтения лекции себя, а то будет не очень удобно. Вот, можно, наверное, начинать.
Хорошо, спасибо большое за приглашение. Мне очень приятно с вами поделиться нашей работой. I will speak English. Hopefully that's fine with you guys. And I'm very excited to be here and tell you a little bit about our computational drug discovery work. Thank you again for the invitation. So I always start my presentations by saying why I'm excited to be a computational biologist and a bioinformaticist today. And one of the reasons is that there are so many publicly available data sets out there that really allow us to ask new questions about disease. And these are just several of these resources. Many of these you're probably familiar with. For instance, the Cancer Genome Atlas is a collection of a number of cancer samples that have been profiled with a variety of different molecular measurements. So RNA-seq, methylation, genotyping, whole genome sequencing. Some of these other resources, the 1,000 Genomes is a fantastic reference set of human genetic variation, as is the EXACT database. Import is a collection of clinical trials data, and the NIH Links is a data set that allows us to look at gene expression profiles and effects of different drugs on various cancer cell lines. It's a fantastic time to be a computational biologist now. This is a slide showing how the cost of sequencing has gone down. This ends at 2014. Now we're in 2021. So it has gotten even cheaper to sequence a human genome since 20 years ago. And also the cost of computation has gone down tremendously. So these devices that we have, our phones, are much, much more powerful than computers were only a few years ago. And it is the intersection of these opportunities that really allows us to create an integrative personal omics profile of an individual. So people are starting to come up with creative ways to leverage technology. Genomics and transcriptomics have been around for a while, but now we can look at antibody repertoire. We can study epigenetics, the microbiome, proteomics. And of course, many of these measurements can now be done on a single cell level, which provides an amazing opportunity to figure out how to integrate these data computationally and also study disease. And this is really what my lab is interested in. We are interested in figuring out how to use data integration to identify new biomarkers for diagnostic purposes. We're interested in figuring out and learning more about the disease mechanisms. And finally, we're interested in therapeutics. And this is really what I'm going to focus on today. And the whole idea behind our therapeutics work is, can we leverage data to find new uses for drugs that are already FDA approved through the process of drug repurposing? So before I dive into drug repurposing, any questions? You guys can hear me okay? Everything is fine?
It's fine.
Yes.
That's fine.
Okay. So the motivation behind drug repurposing is fairly simple. It takes about 15 years and it's incredibly expensive to take a drug, and 90% of drugs fail in early development. One solution to this is drug repurposing, or again, identifying new uses for drugs that are already FDA approved. This reduces the cost of the drug development, but also it reduces the risk of failure. And really, the reason is that a lot of the safety testing for these compounds have already been carried out for their original indication. Does anybody know any examples of drug repurposing?
Ну вот в эпоху ковида дексаметазон стали использовать для лечения ковида, хотя раньше использовали для абсолютно других целей.
Абсолютно правильно. Есть еще идея? All right. Well, most famous and one of the earliest examples of drug repurposing is actually Viagra. It was originally developed for a cardiac indication, and then during the clinical trials, the nurses observed certain side effects, and then it was repurposed. So we sort of want to do the same thing, except we don't want to do it by accident. We want to do these discoveries systematically. So there are many different approaches to computational drug repurposing. There are target-based approaches using genetics, where the idea is if certain diseases share drug targets, then maybe a compound from one disease can be repurposed to another. There is disease-based drug repurposing, or at least I call it disease-based drug repurposing, where the idea is if certain diseases manifest similarly based on their molecular profiles or their clinical data, then maybe a drug from one disease can be used to treat another. And a good example of that is across autoimmune diseases. So for instance, doctors try drugs from one autoimmune disease to another, and sometimes they work and sometimes they don't. There are molecular modeling approaches, so docking-based drug repurposing. And I'll be honest, this is not my area of expertise at all, but there are many groups and companies who are working on approaches to molecular drug repurposing, which really looks at the interaction of various targets and the structures of the compounds and how they work together to see whether certain drugs can be repurposed. And finally, there are network approaches and global signature approaches, where the idea that one drug, one target, one disease is not really sufficient for complex diseases. So the question that we were interested in asking was, can we use public data to systematically predict relationships between drugs and diseases? So we started out with data in the public domain, both drug expression data and disease expression data, and I'll explain what exactly I mean by that in a minute. And we wanted to link the drugs and diseases using gene expression as a proxy. So you can see that we want to come up with some type of a network, like relationship. What we really wanted to end up with is a matrix of diseases by drugs, where each cell of that matrix is a score of how likely a certain drug is to be therapeutic for a certain disease. So what, as a bioinformaticist, what do we do first? We go out and look for data. So the disease gene expression data came from a database called the Gene Expression Omnibus. It's a public data set of pretty much every gene expression transcriptomic study that has either been published or funded by NIH, at least NIH, but also many others. I'm sorry, the numbers are out of date. It actually has over 4.5 million samples currently, and I would estimate, I think, almost 200,000 series. And that 200,000 number is incredibly impressive. That's 200,000 individual studies. And of course, many of them deal with disease. And this includes microarray data, that's older RNA-seq, some single-cell RNA-seq, methylation, and other measurements. But I would say focused on gene expression. On the drug side, we leveraged a data set called the connectivity map. And this is a resource that was developed by the Broad. And what they did is they took human cell lines, cancer cell lines actually, and they treated these cell lines with drugs. The original data set was 164 compounds across almost 500 experiments. And the updated data set was about 1,000 drugs across 1,000 experiments. And they wanted to ask, how do these drugs affect gene expression? So gene expression was measured before and after treatment with these compounds. There's now an extension of this resource called the links. And what they've done there is they actually took over 10,000 compounds, But instead of looking genome-wide at expression, they look at about 1,000 landmark genes, with the idea that the rest of the genome-wide expression can be imputed from those 1,000 genes. The quality of the imputed data is something that we think is potentially a bit questionable. So we've been using the data with direct measurements. So this set of about 1,000 compounds that has genome-wide measurements. And all of this data is publicly available. So our hypothesis was fairly simple. If the disease signature, so the data that we get from the disease, is reversed by a drug, then maybe that drug could be used as a therapeutic. We saw a lot of situations where the expression signatures were similar, and of course, in many situations, we did not see a clear relationship. So we developed a computational pipeline to carry out this analysis. Given a disease signature, we query the drug data, and we come up with a disease drug score. And you can think of this disease drug score as a correlation. If the score is negative, then the drug is likely to reverse the gene expression signature of the disease. So that's the therapeutic prediction that we're looking for here. So this is the results from that initial analysis. The drugs are on the x-axis and the diseases are on the y-axis. Each cell is a therapeutic score, and the negative relationships are shown in green. So we looked at about 100 diseases. We were able to identify a significant therapeutic score for 56 out of them. And you can see a lot of cells here are black, which means there's no significant relationship, which is not surprising. We weren't expecting to find a therapeutic for every disease that we looked at. When we clustered these, we saw that similar diseases were clustering together, and we saw that drugs from similar families were clustering together, which is not surprising. But what we really wanted to do is pick a single disease and see if we can identify a therapeutic for it. So the disease that we chose to focus on initially was Crohn's disease. This is an inflammatory disease of the intestine that has an autoimmune component. It affects about 500,000 people in North America, and there isn't a pharmaceutical cure. The current solutions are to reduce inflammation with anti-inflammatory drugs and corticosteroids, which have pretty bad side effects, and are only reducing the symptoms. And there are surgical solutions, which are quite invasive. So looking at the therapeutic predictions for IBD and Crohn's, we saw that prednisolone, which is used in the clinic to at least alleviate the symptoms for Crohn's disease, was one of the top hits. However, we saw that there was one other drug called topiramate that had a better therapeutic score. So we wanted to investigate that a little bit further. Topiramate is an anti-seizure drug. It suppresses the rapid and excessive firing of neurons at the beginning of a seizure, and it does so by enhancing GABA activation. It's used to treat epilepsy and bipolar disorder. It's an antidepressant, and it has been investigated as a potential treatment for obesity and type 2 diabetes. So we partnered with 2 independent CROs, as well as a group internally that we were collaborating with. This is work done when I was training at Stanford. And we had 4 different animal groups for the experiments that we launched. We had animals that were healthy controls. We used a TNBS model of IBD. So we had the disease model induced that were untreated, animals that had the disease model induced that were treated with prednisolone, and animals where the disease model was induced that were treated with topiramate. The studies went on for a week, and we collected a number of different metrics: clinical signs, pathology scores, histology, as well as endoscopy images on these animals. And it was 4 groups of 12 rats each. So it's a rat model of IBD. This is what the results look like. So this is looking at clinical signs or % of animals with diarrhea. In green, you see the healthy controls. They're doing great. In red are the animals where the disease model was induced. And in blue and purple are topiramate and prednisolone treatment, respectively. you can see that the clinical signs go down significantly with both treatments. However, when we look at the microscopic damage score as well as the gross pathology score, we see a significant reduction with topiramate treatment, which we don't see with prednisolone. And you can see a representative histology image here. So these are healthy. This is the disease model. This is treatment. And also endoscopy images from these animals. When we look at the actual signature that led to this prediction, you can see the disease signature on the right and the drug signature on the left. These signals are not perfect, but at least if you squint, you can see that there is significant reversal. So genes that are downregulated in the disease are upregulated by the drug and the other way around. And when we start looking at the pathways, a lot of them make sense. So you see a lot of immune pathways, IL-15, B-cell receptor signaling, inflammatory response, antigen presentation, and of course, gastrointestinal disease. There is a significant, so these are examples of two genes. Here we only tested a handful. One, TRPV1 is a gene that is downregulated in the disease model and then upregulated by the drug, and IFI30 is the other way around. So this was the original method. Was developed now when I was still a graduate student. That was 10 years ago. And since then, we've applied it to a number of different diseases. So IBD was the first disease. We've applied it to dermatomyositis. Cancer is a very logical application of these approaches because the drugs are actually tested in cancer cell lines. So for some cancers, we can actually match the drug expression data to the disease expression data pretty well. So this is an example of some work that we've done across cancer types with a focus on liver cancer. We've applied it to pregnancy outcomes. But the story that I would like to focus on today is, well, actually two of them that are our most latest applications, one to Alzheimer's disease, and the second story is around COVID-19. So the question that we were interested in asking here is, can we apply this drug repurposing pipeline to Alzheimer's disease? Before I dive into the study, any question on the methods or the approach or anything I've mentioned so far?
There is a question on YouTube.
Okay.
When you talk about genome-wide expression measurement, do you mean RNA-seq or proteomics?
Great question. So the initial study was carried out on actually microarray. So gene expression. We've done this with RNA-seq. Unfortunately, the drug data is older. It's microarray based. But I will talk about this at the end. You can imagine extending these approaches not only to RNA-seq, which is the next logical step, but actually single cell RNA-seq. And that's something that we're working on currently. Great question. Proteomics, you can also leverage proteomics for drug discovery, and we have some ideas around that. We had a study a number of years ago now looking at the correlation overall between RNA levels and proteomics levels across many tissues. And it's very interesting. The correlation is not perfect. Actually, we saw that across tissues, it was ranging between 0.3 and 0.6. So for certain tissues, the correlation is, and for certain genes, the correlation is strong, but for others, it's not. So definitely, it's a limitation. We don't have proteomics data on the drugs. But hopefully, as the technology is developed, this is something that we could incorporate moving forward. Other questions? All right.
The person comments that he's also waiting for a large thermoshift profiling proteomics data.
Awesome. We're on the same page. That's great. Excellent. Okay, so I will dive into the study. This study was published in Nature Aging a few weeks ago, and the work was led by Alice Taubes, who's my first graduate student, jointly with Dr. Yudong Huang. And the work was carried out jointly with Yidong Huang at the Gladstone Institutes. So here, we really wanted to try a precision medicine approach to drug repurposing. And what I mean by that is, studying a complex disease like Alzheimer's disease, in many ways, we think maybe it's not a single disease, but maybe the disease is very heterogeneous. So can we model that heterogeneity in some way? So here we focused on ApoE, and you guys might know that ApoE is the biggest risk factor for Alzheimer's disease. We gathered publicly available data. This is microarray data. It's an older data set, at least to start with. And instead of creating a single disease signature, we wanted to stratify the patients by their ApoE genotype. So we looked at ApoE33, ApoE34, as well as ApoE44 disease. We created expression signatures for all of these subgroups, and this is what that underlying data looks like. So the bar graph here in the middle is showing the distribution of these genotypes in controls and cases. This is a dimensionality reduction, a PCA plot, which is the one in the middle is colored by case control. So you can see a shift, so mostly Alzheimer's disease patients on the left, controls on the right. And this is the same plot colored by genotype. And you can see that there's also a relationship with ApoE genotype, even globally. So when we computed the differential expression, so this is the two Venn diagrams here, as well as the pathway analysis, we saw that when we stratify by genotype, there are some genes and pathways that are common. However, the majority of the signatures are genotype specific. So indeed, ApoE33, ApoE34, and ApoE44 disease are different on the molecular level, at least based on this data. So then we use these signatures against the drugs database that I mentioned, the connectivity map, to look at the therapeutic predictions. And these are the results here on the left. You can see that for ApoE44 disease, so ApoE4 is the risk factor genotype, there was a drug called bumetanide that was at the top of our therapeutics list. So again, we're looking at negative scores here. It does appear to have a negative score in ApoE34 and ApoE33, as well as the unstratified analysis. However, the scores are strongest for ApoE44 disease. And we wanted to investigate that further. This is a graph just showing that shift in expression across the data sets. So bumetanide, or Bumex, is a loop diuretic, and it works by decreasing the reabsorption of sodium by the kidneys. It's used to treat edema and hypertension. And once we identified this drug and thought that it was quite promising, we partnered with Dr. Yidong Huang, who runs an ApoE4 program at the Gladstone Institutes, and launched a number of validation studies. So this is a memory water maze for mice. And the way that it works is that there is a water bath, and there's a hidden platform in one of the quadrants. The animals are released into this container, I guess. And the question that we ask is, how much time the animals don't like to be in the water. So they try to climb out into this hidden platform. So we look at whether they remember where the platform is. So this is a behavior test, and these are the results. In this graph, you can see animals that are 3-3, and you can see that, and shown in black, and animals that are 4-4 are shown in red. You can see that after treatment with bumetanide, and that's this dotted red line here, the ApoE44 animals spend significantly, they're able to perform this test significantly faster and in line with the non-pathogenic animals who don't develop the memory deficit at all. We launched a number of other studies looking at electrophysiology and also validating some of these predictions on a single cell level. I don't have time to get into that today, but I wanted to show you data-driven validation that we looked at using human data following some of these very promising animal experiments. So the question we were interested in asking was, can we further validate these findings using real-world human data? And I'd like to tell you a little bit about leveraging specifically electronic medical records. So at UCSF, we have access to de-identified clinical records for our patient population. It's about 1.3 million patients. And in terms of the data, we have allergies, we have the diagnosis codes, encounters, immunizations, lab tests. We have information on which drugs these patients are on. We have vital information and even imaging data. And all of this data is de-identified for research purposes. So it's the patient information has been stripped from the data. This is showing just the demographics of the population. You can look, it's a pretty diverse population, roughly equally split between males and females. And again, this is the type of data that's available. There's another effort that's led by Dr. Tul Butte, who is my own mentor and also the director of our Baker Computational Health Sciences Institute. And that's to aggregate clinical data across the whole state of California. And that's data on over, this is out of date as well, over 20 million patients, which is quite incredible. So here, we wanted to ask whether there's a lower or higher prevalence of Alzheimer's observed in patients who might be on bumetanide already. So on the UCSF side, this is the left-hand side here, we start with about 1.3 million patients. About 5,000 of them were exposed to bumetanide. We filter some out because we don't have full information on their demographics. And we focus on individuals who are over the age of 65. And then we go through this propensity score matching process where we identify a matched control group matched on age, sex, race, ethnicity, as well as the indication for bumex, so hypertension and edema. And then we look at the prevalence of Alzheimer's in both groups. This is what the data looked like at UCSF. You can see that the patients who are on bumetanide have a significantly lower prevalence of Alzheimer's than the matched controls. We thought these results were incredibly promising. This was work done by Edith Costi in my lab and also Tomiko Oskotsky. And we partnered with a second group at Mount Sinai. This is Benjamin Glicksberg and his team. Mizut specifically was helping us to run the analysis. Mount Sinai is a hospital in New York City. They have data on about 3.9 million patients, and about 3,000 of them had full data and exposed to bumetanide. And you can see that in the Mount Sinai data, we see even a more significant reduction in prevalence of Alzheimer's in the patient cohort on bumetanide. So now we're diving into real world data some more. This is work done by Alice Tang, who's a very talented MD, PhD student in my lab. We're aggregating all of this information, demographics, diagnosis codes, medications, and so forth, to specifically look at the patient population of Alzheimer's patients in the UCSF and cross UC data. This is just a dimensionality reduction plot based on diagnosis codes. So each dot here is a patient. The Alzheimer's patients are in red and the matched controls are in blue. And you can see that there's a significant shift when we look at the UMAP components when we start looking at Alzheimer's versus controls. We're also using this clinical data systematically to look at drugs that might be more common in individuals who are controls, so drugs that might be protective. And it was pretty exciting from our very preliminary analysis to see furosemide, which is also a diuretic, similar class of drugs as bumetanide, showing up here based on clinical data overall. We're also, as I mentioned, looking into leveraging single-cell transcriptomics for drug discovery. This is work led by Stella Belanwu and Yechao Li, who are both graduate students in my lab, looking at creating ApoE-specific, cell type-specific signatures for Alzheimer's using some of the newer single-cell data that has come out recently. This work is available, it's under review, but it's also available as a preprint. So in summary, for the Alzheimer's disease work, we established this ApoE genotype-dependent transcriptomic signatures of Alzheimer's by analyzing publicly available data. We've applied the drug repurposing approach to identify bumetanide as a candidate for ApoE-specific Alzheimer's. We saw that treatment with bumetanide rescued AD-like cognitive and electrophysiological deficits in mice, and we've leveraged single-cell sequencing, single nucleus actually, to explore effects post-treatment in both mice and also in human cells. And finally, to sort of end the validation with human data, we showed that patients who are on bumetanide have a lower prevalence of Alzheimer's that might be associated with treatment from real-world clinical data. So before I jump into and share with you some of our COVID-19 related work that is most recent, any questions about the Alzheimer's work?
I wanted to ask whether the data on patients is available for anyone, or is it open only for researchers of UCSF?
That's a great question. So the patient data is only open to UCSF researchers. You understand that there, of course, even though the data is technically de-identified, I mean, it's still very, very sensitive. So the UCSF data is available to UCSF. The cross-UC, so the data from the state of California, is available to researchers across the University of California campuses. So there are 5 University of California medical campuses, so UCSF, UCLA, UC San Diego, UC Irvine, and UC Riverside, I want to say. Hopefully I got that correct. But it's available to researchers at the individual campuses. But I will mention this. There are now efforts to aggregate clinical data across various centers. And for instance, we partnered with our collaborators at Mount Sinai. We did not have access to their data. We cannot share our data with them directly, but they were able to validate. So we sent them our code, and they were able to adapt it to their data and run the analysis on their side, basically. And there are standards that are being developed to aggregate and kind of centralize clinical data. For instance, one of them is called OMOP. And we've also been able, in a different collaboration that I don't have time to talk about today, to create machine learning models on data in one institution, send those machine learning models to predict, let's say, this was in the context of pregnancy, so predicting adverse pregnancy outcomes, send those models to another institution, run those models there, and actually get performance that's very similar. So I think people are starting to figure out how to share these data, but they are sensitive.
This is just amazing. One more question from YouTube. Yes. Do you see any batch effects across different medical centers?
Yes, we see batch effects in everything. We see batch effects in expression, in any molecular data. Luckily, with the molecular data, we know how to handle them pretty well. As long as your cases and controls are balanced. The situation where you get into trouble is when your cases, so diseased people or disease data comes from one institution or one study, and controls come from another, and then you can't differentiate between the batch and the actual, I guess, biological signal. But in expression and molecular data analysis, there are methods to get rid of batches. For instance, combat is an approach that we use pretty widely. With respect to the clinical data, yes, obviously, the patient populations are different. The clinical care is different. So there's many different differences. However, it's been remarkable to see that some of these signals validate so well. So bumetanide is one example. The overarching analysis that I showed, that we're still pretty early on, the work by Alice. So sorry, I will go back for a second. This, one of her questions is looking at sex differences and how Alzheimer's presents differently in men and women. We actually replicated her findings in the Mount Sinai Alzheimer's cohort, and we could see that about 70% of her findings held up in an independent electronic medical record system, despite all the differences. So I don't know, I wouldn't call them batch effects. They're just differences because it's, you know, again, the patient population is different, environment is different, clinical care is different. There's so many differences, but despite all that, we have been able to validate both individual associations, but also, as I mentioned, machine learning models across institutions.
Thank you so much. One more question is, if we see that patients already having bumetanide treatment have less chance to be subjected to Alzheimer's disease, does it mean that we should use this drug as a prophylaxis?
Great question. We're not there yet. I mean, we still need to run clinical trials to show. This is a retrospective study, right? It's a retrospective question. It's a way to run an in silico validation, but we still need to do clinical trials. And I mean, and bumetanide is a pretty powerful diuretic. So we have to think about the original indication of these drugs and what they actually would do. So it depends. But in any case, additional clinical trials still need to be done before we can answer that question.
We will be waiting for new discoveries.
Thank you. All right. So any more questions before I tell you guys a little bit about our COVID-19 work?
I think.
Okay. No. Excellent. All right. So let me tell you a little bit about how we've been applying both the molecular approaches, but also some of the clinical data approaches for COVID-19. So I probably don't need to tell you this, but COVID-19 has been a huge pandemic in the last year and a half. I'm sorry, this is as of now, not October 2020, but actually November 2021. There have been over 255 million cases worldwide and over 5 million deaths. And there are some treatments that have been discovered, but as we discussed, new drugs have a very long development time, and drug repurposing really came into light in the context of the pandemic. So when this happened in April of 2020, we started asking ourselves, well, what can we do? And the first thing that we ended up doing is applying the molecular drug repurposing approach to some very, very early COVID-19 data. So this work was started and led by Brian Lee, who just finished his postdoc in my lab. Actually, he was a physicist who trained into bioinformatics in my group, a very talented guy. We gathered, three first initial transcriptomics data sets. One was looking at human adenocarcinomic basal epithelial cells infected with SARS-CoV-2. One was an organoid data set from gut tissue, and the very first patient data set, the tiny sample numbers, two patients and 3 controls. This was RNA-seq data. And we computed differential expression signatures. They ranged from a few hundred genes to, in patients, actually almost 1,500 genes that were differentially expressed. We looked at the pathways that were associated across the 3 signatures. And you can see that actually the overlap wasn't, at least on the gene level, was not that big. And this, the pathway analysis was done by Kat Yu, who was a graduate student of my team. We saw pathways that were upregulated, including interferon response, E2F targets, oxidative phosphorylation, and ones that were downregulated, including adipogenesis and mTORC1 signaling. So we took these individual signatures and we created the drug data. And this is showing just the top 15 or so hits across the 3 signatures, the ALV signature, the BALF signature, as well as the EXP signature. And you can see, as highlighted here, that several of them, so the drugs that are shown in purple, were in common. And that was sort of exciting to us. Several of them have been identified by other groups. So for instance, haloperidol and valproic acid, and colchicine was actually in a clinical trial. So we thought that that was pretty promising. We wanted to look at the overlap, and we identified 25 drugs that were common to at least 2 of the 3 signatures that we looked at. And when we visualize them with a network, so this is the drug, this is on the right-hand side, a network. The drugs are shown in these kind of pinkish boxes, and the targets are shown in circles. So the blue circles are single targets, and in yellow, we're showing targets that are shared across the drugs. We wanted to focus our efforts on drugs that reversed all 3 signatures, and there were 4 of those. Vacampicillin was a compound that's used to treat bacterial infection. Clofazimine is an antimycobacterial used to treat leprosy. Haloperidol is a psychiatric medication used to treat schizophrenia. And in terms of the mechanism there, we know that it can bind to sigma-1 and sigma-2 receptors, and has also been shown to decrease viral growth in SARS-CoV-2 infected vero cells. Valproic acid is used to treat epilepsy. It's a HDAC inhibitor, and previous studies have not shown it to be efficacious. So we launched 2 independent validation experiments. One was in collaboration with Melanie Ott and her group, and this is looking at CALO3 cells. So these experiments were all carried out in cell lines. And we saw that haloperidol had this very nice dose-specific response. So here, this is the controls. These are cells that were infected with SARS-CoV-2. You can see that remdesivir gets rid of the infection, and you can see a significant reduction with haloperidol as well. We launched a second independent study with collaborators at Mount Sinai. This is Adolfo Ssris group and Chris White. And here we tested a number of the compounds. And these graphs are showing percent infection in red. So we want this curve to go down, while we want the cell viability curve to continue, cells to be viable. So we want it to be straight. And you can see that for a number of these drugs, vacampicillin, cyclosporin, clofazimine, we see that effect. And here's a bunch more that we tested. So this was kind of a high throughput testing. And of course, we had remdesivir as a positive control here. So in terms of the summary of this molecular application is we developed a computational drug, we used our computational drug repurposing pipeline on many different diseases, including COVID-19. Here we identified 25 hits that were common across at least 2 of the signatures and 4 consensus hits. And then we had 2 independent validations showing at least some efficacy of viral inhibition in 11 out of 16 common drug hits and 3 out of 4 of the consensus hits. And this work was published in scientific reports earlier this year. So if you're interested, I will share these slides with you guys so that you can have the references as well. So in the last few minutes, I wanted to tell you about some of the most recent work that we've been doing leveraging clinical data. And you were asking before about aggregating and sharing clinical data. And this is an effort that I'd like to highlight, carried out by the Cerner group. So what they've done at the beginning of the pandemic is they started aggregating clinical data and making it available for research purposes. Our group was incredibly lucky to get access to this electronic medical record data. So the data that we worked with included 500,000 COVID-19 patients across 87 hospitals in the US. It's an incredible amount of data. The population roughly equally split, a bit more females than males, And the data that was available included demographics, medications, procedures, encounters, diagnoses, lab test results, imaging data, and past medical history up to 5 years for some of the individuals. So incredibly rich resource. We had to apply to get access to this data, and again, we were lucky to be able to work with this. So there were several questions, kind of hypothesis-driven questions that we wanted to ask. The first one was looking at statins. As you know, statins primarily are used for lipid reduction. They have known anti-inflammatory and immunomodulatory properties. And there have been some evidence for effect of statins and COVID-19 to showing reduced disease severity and reduced mortality. So we partnered with a group at Stanford. So this is a collaboration with Ivana Marek. The person on my team who led the analysis is Dr. Tomiko Oskotsky, and also my co-senior investigator on this is David Stevenson from Stanford. And this work was published earlier this year. And what we've shown is that consistently, using a similar schematic of identifying a patient population as well as a matched control group based on demographics and other clinical parameters, we saw that patients on statins had a significant reduction in mortality rate. And this is showing data across 10 independent iterations. Most recently, and this has just got published earlier this week, we've shown that another set of drugs, and specifically SSRIs or serotonin reuptake inhibitors or antidepressants, reduce the risk of mortality in COVID-19 patients as well. So this was just published in JAMA Network Open. The idea was quite similar. We started out with patients from the Cerner database, almost 500,000 patients, focused on those with a, so these are all individuals with a diagnosis code of COVID-19. However, we wanted to focus on patients who had a laboratory test confirmed and ICD-10 code. So that's about 100,000 patients. The majority of them had complete demographic and were adults. About 3,000 of them were exposed or were on SSRIs during the defined time frame of their COVID-19 diagnosis. And you can see the breakdown. So specifically, we focused on patients on fluoxetine, which is Prozac, fluoxetine or fluvoxamine, or any SSRI. We carried out a similar propensity score matching strategy, looking at exposed versus unexposed individuals. And we were able to show that there was a significant reduction in mortality rate. So this is work led by Dr. Tomiko Oskotsky. And to summarize, we were able to show that overall there was an 8% reduction in mortality among patients on any SSRI. However, the largest reduction of 28% of relative risk of mortality was for patients treated with Prozac. And again, this literally just came out this week. Why SSRIs? You know, our study just identifies an association. It's a demonstration of how you can use data to test a certain hypothesis. But there have been a number of different studies that have shown that there might be links between serotonin and the immune system, that fluoxetine. I will summarize again. What we're doing now, I've shown you some ways that we leverage molecular and clinical data for drug discovery. So what we're working on now is exploring combination therapy approaches, applying more advanced machine learning, incorporating single cell measurements into drug prediction, and leveraging electronic medical record data to identify new uses for existing drugs. And I'm hoping that by integrating clinical and molecular data, we can get a little bit closer to precision medicine. So with that, I'd like to thank my group. The Alzheimer's work was led by Ali Taubes, and now carried out by Stella and Yachal on my team. The drug repurposing for COVID-19, the clinical analysis has been led by Tomiko Oskotsky and Brian Lee and also Guy Androletti. We've had a number of collaborators across many of the pretty much every single study that I mentioned. So for Alzheimer's, we work very closely with Yidong Huang and his team. For COVID-19, Melanie Ott, Adolfo Garcia Sastra, Ben Glicksberg at Mount Sinai. And this is my team. This was us in February of 2020, where we could all pack into a single elevator. And for the past year and a half, we've been mostly on Zoom. We do get to see each other once in a while, which is wonderful. And I'm hoping that things in that department will get better. With that, I would love to take any additional questions. And this is my email and lab website, in case you guys want to learn more. So I will stop sharing. And I would love to take any questions.
Thank you so much for the amazing lecture. It was very interesting. And I wanted to ask, as the pandemic is going on, do you have any plans on integrating SSRIs in clinics?
Yeah, well, again, with SSRIs, just like bumetanide, still clinical trials need to get done. There was a small clinical trial looking at SSRIs, actually, and it's shown some benefit, but we still need more clinical trials before this can be, used in patients. But it's promising because the drugs are so cheap. The drugs have been around for a long time, so hopefully it's something that will be studied further, and we're going to be working on that as well. Other questions? I know we have like 2 minutes.
Подождем еще минутку, наверное, вопросов на Ютубе, потому что там немножко отстает трансляция.
Хорошо. Плюс было плохо слышно.
Хорошо. Плюс было плохо слышно. Хорошо. Плюс было... Кажется, вопросов больше нет. Вот появился вопрос. Do you have plans to leverage some general graph-based ML to identify common molecular features of drugs with the same effect?
Yeah, it's a great question. I would love to. I'm not an expert in graph methods, but we're starting to look at ways to integrate different types of data, and especially with our efforts to look at combination therapy using single cell approaches. I think some of the applications of graph approaches could be really helpful there to look at which, if you build a network of drugs based on structural similarity or molecular similarity, can we identify pairs of compounds that might reverse a gene expression signature better? Absolutely. Other questions?
People wish you good luck and thank for a really nice presentation.
Thank you. Thanks for having me. It's my pleasure.
Спасибо большое, Марина. Спасибо большое за лекцию.
Не за что, я рада.
Спасибо
всем. Новых открытий и приглашаю рассказывать о новых прекрасных находках.
Спасибо.
Спасибо всем, кто сегодня был с нами. Запись сегодняшней лекции скоро появится на Ютубе. Всем спасибо и до новых встреч. Еще раз, Марина, спасибо вам огромное.
Спасибо вам. Всего доброго.
Всего доброго.